Hey, welcome to the first slide show on blood. So begins AMP2. We're going to talk about blood for quite a while. And we're starting here by looking at something that's not human, some cells that are definitely not human cells. These are bacterial cells. I have no idea what kind they are. You got to ask a microbiologist for that. But they're living cells. So they need oxygen. They need nutrients. They're got to get rid of wastes, carbon dioxide. And the question we got to answer is, how does that stuff come in and how does that stuff get out? And the answer is something you learned about quite a bit in AMP1. It's the process of diffusion. Stuff diffuses in and diffuses out. Now, here's the thing. There is a problem with diffusion. There's a limitation on it. And the big limitation is it isn't good over large distances. So size is a problem. And with that, we gotta realize that you and I, and this baby elephant here, we are big animals. We cannot merely rely on diffusion since we have 75 trillion cells or so. We need a circulatory system. That's what we've got to have. And any circulatory system has got to have three parts. It's got to have some sort of pipes, blood vessels in this case. It's got to have some sort of pump, which of course is going to be the heart in this case. And it's got to have a fluid, a transport medium. And we've got all three. We got all three here. All three basic components. We got our pipe, pump, the heart. Got our pipes, the blood vessels, the arteries and the veins. And our fluid is blood. And that's what we're talking about today, blood. The fluid connective tissue that's within our circulatory system. Blood consists of these cells. I'm using the word with air quotes. You can't see the air quotes, but I'm making them right now. These cells that we refer to as formed elements, because they're not all exactly cells. That's why I put the air quotes out. And these cells are in a liquid called plasma. So on the screen here, we do see a lot of red blood cells. They look like empty little pink donuts. See a couple, well, not a couple, we see 4 white blood cells as well. And in the left-hand corner, kind of underneath the word formed, we see a couple little purple-ish platelets. So let's talk first about blood in general, some physical characteristics of it. It is either red or red. It's bright red, a scarlet, if you will, if it's rich in oxygen. It's a darker red if it is poor in oxygen. It's pretty viscous compared to water, about 5 times as viscous as water. Now, viscosity is something where you got to have some of it. I mean, blood's got to have stuff in it. It doesn't work unless it's got stuff that's being carried around, it's being circulated. But if it is too viscous, it's like honey, for example, it won't flow very well. So you need to have a good balance there. And of course, you know what that balance, the term for that balance in our body is. I know you do. All right, volume-wise, about 4 or 5 liters or so. So, 2.5 of those 2-liter bottles that you're familiar with. Temperature is a little bit greater than what we consider normal body temperature. Temperature is about 100 degrees, a little over 100 degrees Fahrenheit. And that's because blood is carrying heat. All right, real quick, let's talk about the pH of blood. As you well remember from chemistry class, pH is a measurement of hydrogen ion concentration. Mathematically, it's the log of 1 over the hydrogen ion concentration. And the thing to take away from this is that if you have a lot of hydrogens, then your pH is going to be very low. And a low pH is acidic. If you don't have a lot of hydrogens, your pH is going to be high, and a high pH is basic. So if you're right at 7, you're right perfectly in the middle, you're neutral. If you're less than 7, you're acidic. If you are greater than 7, you are basic or alkaline. All right, check this guy out here. This is George Washington. You probably recognize him. On his deathbed, we're going to talk later on about how he died. All right, I want to talk about that in the lecture period. With that, Let's move on to the functions of blood. It's going to distribute stuff. For example, gases, oxygen, carbon dioxide. Nutrients, glucose, amino acids, fatty acids, vitamins, lactate. I could go on. Wastes, urea, other nitrogenous wastes, bilirubin. Electrolytes, sodium, calcium, chloride, etc. Water, signals, hormones, and there's more. It's going to help with regulating things by moving heat. It's going to regulate body temperature. By moving hydrogen ions, it's going to help regulate body fluid pH. By moving water, it's going to regulate body fluid volume itself. Protection is something we also have to consider as a function of blood. And what we mean by protection here is protection primarily against pathogens, against disease. And this is done by the virtue of having white blood cells and immune proteins in the blood. All right, let's keep moving. So, got a little story about a man named John here. And he's really tired. He feels like his muscles lack energy. And of course, you know the basic cellular form of energy is ATP. So he decides to go to see a doctor. There she is. And she's going to check him out. Now, in order to make ATP, what do we need? Well, we need some chemical form of energy. We need perhaps some glucose. We need perhaps some oxygen. And John gets some blood drawn. Blood is drawn in a syringe. We see it there. It's spun in a centrifuge. And a centrifuge is going to separate components of blood by their density. And once the blood is separated, you're going to see three distinct layers. There's a fluid layer up top, which is called the plasma, a liquid layer. And then near that, not beneath that, I guess, there's a layer called the buffy coat. Buffy, it refers to a color. There's a color called buff. Supposedly that's the color of buff in that right-hand corner up there. In the buffy coat, we've got white blood cells and platelets. And notice how small it is, that little buffy coat. Sometimes you can't even see it with your naked eye. At the bottom, we got a big red area. And that's where we find our red blood cells. Now, what I want you to think about is which one of these three areas might be problematic for John. If you need to, look up what these three blood cells, these three formed elements, red blood cells, white blood cells, and platelets do. And think about which one of these is going to be a problem for him. All right. Now, if we're measuring hematocrit, which is a measurement of that we can get from this centrifuge blood, hematocrit is the percentage of whole blood occupied by red blood cells. So what you would do is you would take that centrifuge blood, you would measure how high the red blood cell portion is, or how tall, long, whatever word you want to use, measure how long the whole blood sample is, divide the red blood cell height by the whole blood height, and then multiply that by 100 to make it a percent. Normally for a guy, it's somewhere between 42 and 56%. For a lady, it is between 38 and 46%. Maybe you can think about why a guy might have more red blood cells. It is a hormonal reason, and I'll ask you about that in class as well. All right, on this screen, I have a little practice for you from, if you've done the lab on blood typing and hematocrit already, you can do this. If not, wait till you've done that. And what I want you to do is figure out which of these simulated hematocrit tubes is most likely to be John's. All right. Here we see some of our formed elements again. The red arrow at the top is a red blood cell, also known as an erythrocyte. And the basic main job of a red blood cell is to carry oxygen. Upper left-hand corner, black arrow. those are platelets. And platelets are little cell fragments. They play a big role in blood clotting. The other large cells that we see here, the blue arrow indicates one. You can see 3 others as well. Those are white blood cells or leukocytes. They play a big role in immune defense. And all of these formed elements are floating in a liquid, which is known as plasma. So plasma is the liquid that contains the blood cells. And, excuse me, plasma is 90% water. And water is great because it is a wonderful solvent. So a lot of things are dissolved in here. All right, things like many of the things I mentioned earlier, gases, nutrients, electrolytes, wastes, they're all dissolved in there. So we're going to have electrolytes. We see some samples up there. Nutrients, whole vegetables, not exactly. Amino acids, simple sugars, fatty acids, other assorted nutrients. Wastes as well. Nitrogenous wastes, urea, ammonia, that sort of thing. What else is dissolved in there? Well, vitamins are going to be dissolved in there. Some of the vitamins, it's kind of a problem because they're fat soluble. So vitamins A, D, E, and K are actually going to need a special protein boat to ferry them around. Cholesterol, the molecule up there on the left, is in a similar situation. We need it to be transported in our blood, but it's hydrophobic. It's fat soluble, not water soluble. So it needs help being carried as well. All right, let's talk about some other stuff we find dissolved in blood, including the plasma proteins. There are a lot of plasma proteins. We're going to meet several of them over the course of the semester. The most abundant one is albumin. It maintains the POP or the plasma osmotic pressure. It also transports steroids. Think about the normal steroid hormones that you have in your body. Also transports a red blood cell breakdown product called bilirubin that we're going to talk about more a little bit later. Now, albumin is made by the big brown organ in the picture here, which of course is the liver. Now we know albumin maintains the osmotic pressure in your plasma. So a little review here, and we got to consider a little situation. Here we got 2 solutions that are separated by a semi-permeable barrier, and water can pass through the barrier, but red particles cannot. So my question is, which way will water flow? In other words, you got to figure out which side has the greater osmotic pressure. And osmotic pressure is the one with the greater osmotic pressure is where water is going to go. So I'm going to ask you about this in the lecture period, okay? And then we're going to talk about quasi or core as well. I'm going to ask you about this, so come prepared. All right, other plasma proteins. We've got the globulins. Globulins are primarily used in transport, alpha and beta. Now I know that's not an alpha up there, that little symbol. I don't know why it came up like that. So alpha and beta globulins transport things like metal ions, lipids, and the fat-soluble vitamins A, D, E, and K we mentioned before. We also have things called gamma globulins or antibodies. These are immune proteins made by these special cells called plasma cells during your immune response. All right, let's take a look at a bacterial cell here. And we see an antibody binding onto it, an immune protein binding onto it. And what this is going to do is allow the other cells of the immune system to recognize and destroy this bacterial cell. Okay, one last slide. We also find in our plasma buffers. Buffers are chemicals that resist changes in pH. We need the pH of blood to be maintained between 7.35 and 7.45. two major buffers in our plasma are albumin, which we mentioned already, and bicarbonate. We got a little question for you here at the end in the green down there to consider. We'll talk about that later. See ya.